Okay, so this video is going to cover leukocytes and probably platelets as well, because the leukocytes we go back over in another chapter on lymphatics. So the leukocytes, as shown here, right, there's 5 different types, so these are actually 2 ways of looking at the monocytes. So they defend and protect and clean up the mess. So we've got the fills are all going to be able to be phagocytic. So do pay attention to that. And they'll also be called the granulocytes, and these will be the agranulocytes. Leukocytes are true cells. They have organelles, and they certainly have this nucleus. In fact, I tell students to tell the difference between them by looking at the appearance of the nucleus. As you can see here, they're larger than the red blood cells, though lymphocytes can sometimes be very close in size. There are actually a lot less white cells, so this is, like I said, a photoshopped situation. These cells will be able to leave the blood vessels because, think about it, most of the time you're not defending yourself solely in what's in the vessel, but out in the tissues as well. So this shows you the reason that they would need to leave the red blood cell, right? The blood vessels. So here we are in circulation, and you've been cut, and you're being infected by some sort of bacillus bacterium, which is collectively called the pathogen. And the damaged cells, and so here's our epidermis and dermis, are releasing these paracrine and substances, which are calling, if you will, we call it hemataxis, the white blood cells. So they are moving through the blood vessel, and when there's an increased concentration of these paracrine substances, they will try to leave, right, the blood vessel. And this is assisted because these substances also make the blood vessels a little bit more permeable or leaky. So the spaces between cells becomes a little bit greater, and blood flow increases, so that delivers more white blood cells, and they leave. And of course, they leave to defend you. All right, so again, neutrophils and monocytes are the most active in this because monocytes get to be very, very big, and so they can consume a lot of bacterium. And neutrophils, by default, are the most numerous in there, so they can certainly, being so many of them, they're going to be able to defend you against so many invaders. All right. I won't use the word diapedesis or chemataxis on a test, but that's what the word. But diapedesis is a process by which the white blood cells distort and push their way through, and therefore out of the blood vessel. And then chemataxis is this idea of being called. All right. So the 2 categories, again, we talk about this lab, granulocytes and agranulocytes. So agranulocytes without granules. And remember that those granules that you're seeing, even the ones you aren't seeing that actually are present in these monocytes and lymphocytes, are assisting these cells in what they do. So cells are bags of chemicals. Even though red cells were bags of chemicals, it's just they just had one chemical. Right? So if you're a granulocyte, your nucleus will always be lobed. So it's one nucleus, but it appears as though there's multiple. If you're an agranulocyte, you have this lovely round one as a lymphocyte, or the kidney bean shaped or horseshoe shaped. If you are a granulocyte, and they all end in the term fill, right, these 3 here, you're going to be about the same size. So size isn't a way to tell the difference. If you're an agranulocyte, monocytes are the largest, and lymphocytes tend to be the smallest. We then distinguish the individual granulocytes, which all end in fill, by the color and size of the granules in their cytoplasm. Now, all of these are made in the bone marrow. All of them are able to consume other cells, and here we're talking mostly bacteria, viral particles, right? And but have different chemicals, so serve slightly different purposes in your defense. If you're an agranulocyte, again, no visible granules are in your cytoplasm, but you are also going to defend in various ways. So the next couple slides just really go through and talk about each of the individual cells. So again, the neutrophils have, this is a higher magnification than we can achieve with our microscopes, but have these tinier granules. They have multiple lobes, so 3 to 6, whereas the other 2 fills have only 2, generally 2 lobes. So these are multilobular. In fact, they call them polymorph, so many shaped nuclei, and hopefully you've seen that. They're very good at defending against bacteria. As we said, they're eaters, and fungus, and they, again, eat that. Their granules actually are very powerful chemicals, hydrolytic enzymes, so destroying things. So hydrolytic hydrolysis is a degradation chemical process. Defensins, which are going to poke holes in membranes of other cells. Oxidizing substances, which is similar to bleach and hydrogen peroxide, and then they even have lysozymes, which are these are chemicals, enzymes specifically, that destroy the cell wall of bacteria. Right? Eosinophils or eosinophils, they have generally only two. That's what the bilobed nucleus. These have these very larger red-orange granules, which are basically antihistamines, so they're going to help minimize your allergic response, and toxins to parasites. And that's why you see these increase when someone has been invaded by a parasite. Okay, basophils, the largest granules, and these are histamines and heparin, and they're going to aggravate your allergic response. But it's really, they're there to help promote inflammation, which is a defense response you have when you have been invaded. However, oftentimes, like I said, when you have an allergy, that's an overzealous or out of place immune response, and so they end up making your life kind of difficult. All right. So, but heparins and histamine, and this causes dilation, so more blood flow, and this is also going to attract other white cells. And these are the ones I say are easy to mistake as a lymphocyte, just because the lymphocytes have these nice round, consistent nuclei, and you see a basophil and you think, oh, that's a nucleus, but no, it's the whole cell. So they are the rarest, so that's why I said you probably didn't see any with your microscope. Agranulocytes, or the agranulocytes, are lymphocytes and monocytes. So here again, a lymphocyte close to the size of a red blood cell. There's quite a few of these in circulation. They can remain long periods of time. Many of them, though there's a lot in circulation, are found actually in your lymph nodes, your spleen, thymus, tonsils, and so forth. We are going to look at the three groups of these later on when we talk about that. And again, this is there to clarify, here's what you have to look forward to as far as their role in your defense. And they're going to be responsible for the specific immune response. Right? The monocytes are the largest, as you can see, compared to this red blood cell. Their nucleus is shaped like a kidney bean or a horseshoe. When they leave the blood vessel, they actually change into a much larger cell known as a macrophage. And they're really good at consuming dead, dying cells and invading cells. Right? So this gives you, again, a table to show the different types, groups them together, the numbers, which you won't have to know from me, and characteristics, function that they serve, and a little bit more characteristic of them. But this is a pretty thorough table, which is a good reference for you guys. But again, the only thing I really am going to ask you to know specifically is that, and I've mentioned this before, when I said leukocytes and their production are controlled by interleukins and colony-stimulating factors, which are types of cytokines. All right. If you produce too many, know that the term is leukocytosis. If you don't have enough, it's leukopenia. And then I give you a variety of reasons that this might occur. Right? So the platelets, right, thrombocytes, are very, very small. They're quite a bit smaller than red blood cells. It turns out they're just pieces, right? So what happens is in the red bone marrow, right, as the hematocytoblast differentiates through the myeloid to the megakaryoblast to megakaryocyte, it gets so big because it's repeatedly undergoing the cell cycle in mitosis but does not do cytokinesis that it can't move out of the bone marrow, and so it extrudes or extends its processes, part of the cell, out, and it splinters. And so you get platelets from that splintering, okay? Again, here what I really want you to know is that the chemical that stimulates the production of platelets is called thrombopoietin, all right? That these platelets are also banks of chemicals, and these chemicals are going to help in the process of clotting that we'll look at. If you have too many platelets, we call it thrombocytosis, so again, similar suffix. And if there's too few, it's thrombocytopenia. So, all right. We're not going to do the Kahoot here. If you guys want to do a Kahoot, I do need to send you the link, but that's this is our stopping point.